Hello, this is Dr. Yunong Liu. In this short video, I'm going to talk about the public resources for NGS data. So the first resource I want to talk about is the Thousand Genomes Project. And this is a very famous project that probably most of the audience already know, heard about it. So essentially, initial goal is to sequence 1,000 individuals' whole genome. If we think about the Human Genome Project, which is ended in 2003, the purpose of Human Genome Project is to sequence the one draft of human genome. So trying to understand what are the sequences that define a human being. But the Thousand Genomes Project is an extension of that. It's trying to understand the variabilities across different individuals, both within individual ethnic groups or across different ethnic groups. So eventually, at the project being completed, they sequenced 2,500 individuals from 5 major ethnic groups. So the list is listed here, and it's about 500 for each individual group. So this project not only gave us very, very useful data for biomedical research, but also generate a lot of data tools as well as the standard. So some of the selected results out of that. And from the Thousand Genomes Project, people identified over 15 million SNPs across 2,500 individuals. And there are about 1 million short insertion deletions being identified, 20,000 structure variations. And also they found that each person carries about 250 to 300 loss of function variants in annotated genes and 500 to 100 variants implicated in inherited disorders. So these are very interesting data that we otherwise will not know, because when they really select individuals for Thousand Genomes Project, those individuals are actually healthy individuals. And this is the first time that we know there are a lot of genetic variants in those as well. So the second project is also very useful, is Genotype-Tissue Expression Project, or GTEX project. So the purpose of this is to provide a reference data set to study genetic changes and the gene expression. If we think about studying DNA, to a certain extent, it's easier because our DNA, other than special situations like in cancer situations, our DNA are the same across all the cells in our body. However, for RNA level, that will be a different story because every tissue type will be different, and every disease conditions could be different. So the question here is, how do we define normal for a specific tissue type for the RNA level? So this project is trying to address that goal. It's launched in 2010 and just being completed not long ago. And eventually they sequenced over 11,000 samples across more than 700 donors, and each donor has multiple tissues. So totally across 53 different type of tissues. So this project also gives us a lot of information regarding the RNA reference data sets. So it's very useful. We are currently still using that a lot. So there are many other democratic statistics available that in the website I'm listed here. So using this data, there are many major outcomes in addition to serve as the reference for everybody else to use. For example, it provides a reference for expression level across tissues. And people also studied expression quantitative trait loci analysis using these data sets, essentially trying to understand how the genetic variants at individual gene level that is associated with the gene expression. So they identified over 19,000 cis EQTLs and close to 100 trans EQTLs. The data are all in the public domain, and the publication can be found in this Nature Collections in November 2017 issue. and the processed data are available from the GTEX data portal, and the raw data is actually for the original sequencing data. People can find that in the dbGaP, which we will introduce dbGaP later as well. Next project is ENCODE, or Encyclopedia of DNA Elements. The purpose of this project is to annotate the genome. So if we sequence the individual, we can easily get 3 billion letters that represent the DNA sequence of this individual. However, which part is corresponding to what? For example, a certain part corresponding to a gene, which part corresponding to exon regions, intron regions, promoter, enhancers. So the ENCODE is trying to address this question by annotating the genome. So they're using a huge number of different technologies, all the way from the experimental-based RNA sequencing, ChIP-seq, CLIP-seq, DNA-seq or ATAC-seq, Hi-C or other type of chromatin conformation assays, as well as the computational predictions. If you want to access the ENCODE data sets, actually in the UCSC Genome Browser and WashU Epigenome Browser and many other resources, they already integrated ENCODE findings in that, so you probably don't have to process the data. However, you can also download the raw data to do the processing. Here are the publication from the Nature issues, as well as the data access and the tracks are available in major genome browsers. Similar as ENCODE project, there is a parallel study called the MODE ENCODE, which study the model organism for the gene annotation, and they focus on the Drosophila and the C. elegans. So major findings has already been published as well. There is another consortium called Epigenomics Roadmap Project. It was developed in parallel with ENCODE, which initially focused on the cell lines, and an epigenomic roadmap project focused on the stem cells as well as tissue types. And they focus on epigenetic features, including the DNA methylation, histomodification, chromatin accessibility, as well as small RNA transcripts. And major data are available as well. I want to mention here that currently ENCODE project also has is still continuing, moving in addition to the cell types, the cell lines they're using, they are moving into the tissue types as well. Another very famous consortium is the TCGA, or the Cancer Genome Atlas project. The goal of this project is really trying to understand the molecular features of different types of cancers. And in this project, they actually focus on the 33 different tumor types. And they sequenced the tumor samples out of 11,000 patients. And they are not only sequenced the DNA or RNA, instead, they were trying to sequence many different types of molecular features out of these tumor cells, including the DNA part, which identified germline and somatic mutations, copy number variation, as well as structure variations. The RNA level, including the messenger RNA, total RNA, and the microRNAs. And also there's DNA methylation as well as proteomics data. All the data are publicly available for tier 3 and tier 4 data that you can very easily access in the public domain. And for the tier 1 and tier 2 data, you do need to have special permission to download the data from other sources, which you can get through the Genomic Data Commons data portal system, and as well as visualizing data with the CBio portal. In this module, actually, Dr. Kun Huang is going to introduce the CBio portal in a much better detail, and he's going to introduce the theory part of it, and also attach several YouTube training videos on how to use the CBio portals. Similar as the TCGA project, there was an international effort of doing very similar goal, understand the different molecular features of the cancer, and eventually TCGA become part of this international effort or ICGC consortium. The data is also publicly available. Another project or consortium that the data is available and we use that very regularly is called the gnomAD, or earlier phase is called the EXAC project. The gnomAD is a genome aggregation database, or EXAC project is exome aggregation consortium. So you can see that EXAC is the initial phase that focuses only on the exome sequencing data, and later they were being extended to whole genome sequencing data as well. So the purpose of this project is really trying to harmonize the data that is already in the public domain. We know that there are many different sequencing projects at different levels are being supported by NIH or other funding agencies, and there are many different consortium being developed. So the data we already talked about, there's 1,000 genomes, the GTEX, the TCGA, and there are some of the, for example, ADSP or Alzheimer's disease sequencing project, and there are many such projects. But when people are trying to use this data, sometimes we run into trouble because we have to download the raw data, we have to somehow process the data in the uniformed way, and then integrate the data together. Well, for this project, they were trying to do this for us. So basically, they got the data, they aggregate them together, harmonize in the uniformed way, and process in the uniformed way. And then, so you can readily download the VCF files that generated, tell you the genetic variation of the individuals. So data is very, very useful and is publicly accessible. Another consortium is called the Cancer Cell Line Encyclopedia or CCLE project. From the name, you can say this project really focuses on the cancer cell lines. So this started from many years ago, about more than 10 years ago, and their goal is to find out up to 1,000 different cell lines, and they were trying to understand the genomic features as well as the transcriptomic features of these cell lines, including the DNA copy number variation, and initially they use Affymetrix SNP array, later move on to the sequencing, and then they also measure the mRNA sequencing levels. So if you got a cancer cell line, you want to know what the gene expression level is supposed to look like, and this will be a good resource you can get. They have the data in the raw data form that you can download and further process, or you can get the data that on the processed data that is already available there. So there are actually a sister project of this CCLE, because CCLE only focus on molecular data, the RNA level and the DNA level data. And we don't know how these cancer cells actually behave. So there is a sister project called the CTRP, or Cancer Therapeutics Response Portal. And the idea is they take all these cell lines and they treat these cells with different drugs or compounds. So you can see this, the CTRP data has over 480 compounds treated on 860 cancer cell lines, and they will see this response. That's a huge amount of phenotypic characterization of these cell lines. Sometimes it can be very, very useful for us to study these cancer cell lines or use these cancer cell lines. Very, very important resource. And actually my lab had a student that is working on that and had a publication recently out of it as well. Similarly, there is another cancer cell line related project called the LINKS program, NIH LINKS program, which is a library of integrated network-based cellular signatures. So if you think of the CCLE, they give you the gene expression data, but that is a baseline level data. They treat the cells with different drug, but they are not going to measure the transcriptome features after the drug treatment. They only tell you how the cells behave in terms of the survival or all those drug resistance and sensitivity informations. But before and after drug treatment, with different doses, what are the expression level change? That is something that they have not measured. But this LINKS project is trying to address that. So if you think about 100 cell lines, 1,000 cell lines, several hundred drugs, that is a huge combination. And to get all the global gene expression levels at that level of all the different time points, that is almost not approachable. So what they are doing is a very heavily barcoding or multiplexing approach. Eventually, they only measured 1,000 genes for every single cell line in every single condition, which makes it very, very cheap. But they can profile many, many different conditions. So in this 2017 Cell paper, they actually published 1.3 million public accessible profile data. That is a huge amount of resources people can use. The next very, very important resource is the GEO database, which is the Gene Expression Omnibus. And this is where you should go there to look for some of the published studies. And if you want to look at their raw data, normally this is where the data are being deposited. And they have a very nice system, not only to put the raw data in, but also provide the documentation for the experimental conditions, the treatment, the reagent being used, the bioinformatics, the processing methodology, and so on and so forth. So it's very useful. And we are going to put in one training video of this to give you a general introduction of the GEO database. And that video is done by one of my students, Chuanpeng Dong. There is another similar resource, or called EBI Expression Atlas database. It's very similar to GEO, but that is most on the European side. In most of cases, most of the major publications, when you publish RNA sequencing data, they will request you to put your raw data and the processed data into some of the public repository, and GEO is one of the to-go places that most people will select. Well, the GEO is not a protected data, which means that you get you can have cell lines, you can have animal models, you can have many different type of molecular profiles. However, one thing you cannot put in is the sequencing data, either RNA sequencing or DNA sequencing, whatever sequencing level data coming from individual humans, because those are the information that needs to be protected by HIPAA law and other laws. So you cannot really just dump it in the public domain that everybody can have access to. For those type of data, the molecular data or sequencing data coming from individual patients, those data are normally being deposited into the dbGaP project database, which is the database of genotype and phenotypes. And they have all different levels of molecular data, including the DNA level, RNA level, epigenome level, and the somatic mutations, germline mutations. So they have over 2.4 million different assays. It's a huge amount of data there. And they also have phenotype data of these individuals. It's very, very important resource. But in order to utilize the information in this database, you will have to do some paperwork in terms of routed through your university institution and making sure that these patients, the privacy and everything is properly protected. The last resource I want to go through is about a microbiome project. There is a NIH Human Microbiome Project, and we haven't spent too much time on this for this lecture series, and maybe we want to add in 1 or 2 lectures on that. But the point is, in the human body, there are a huge amount of microbiome species. Actually, by the number of cells, they outnumber human cells 10 times. So, well, how many cells we have that in the human body? There are 10 times more bacterial cells that are on our body. But there are smaller cells, and but they are about normally 1 to 3% of the body mass are microbiomes. And actually, from the genetic diversity, they are 1,000 times more genes than human genes because there are so many bacterial species out there. And more recently, that microbiome, the importance of a microbiome in the human body become very, very important. Now we know that almost every single major disease are related to microbiomes. And of course, diabetes, cancer, obesity, GI disease, lung disease, those are. And now we recently know that Alzheimer's disease and many other diseases are related to the microbiomes. So there is a NIH consortium about human microbiomes project, and there were 2 phases. And the first phase is really trying to understand the microbiome reference for healthy individuals. It's very much like the GTEX project for the RNA level provided reference, or 1,000 genomes at the DNA level for healthy individuals. This one is for healthy individuals about their microbiomes. So they select over 300 healthy individuals across many different body parts, and then they get the samples and trying to identify what are the microbiome species on that. So the second phase is more disease focused, including the pregnancy and preterm birth, IBD, and type 2 diabetes. And this is a booming area, and many great effort are being put in there. So you can visit that website I'm listing there, and all those data are in the public domain. So this is all I want to talk about today in terms of some of the selected public resources for genomics or NGS data. And all the data I talk about are publicly available. And these are the resources actually in our labs. We are using that very routinely to answer biological questions that we have. Okay, and in this module, we also have 2 resources. We'll tell you a little bit more detail. One is the GEO database. The other one is the CBio portal. And stay tuned. I hope you enjoyed that. I will see you this Friday.